Okay, so second half of the talk then, I'll give you, again, a pretty high overview of the company that we set out in effectively 2020, just in time for the pandemic. Great timing. So yeah, I guess we're Green Bioactives, pioneering the discovery, development, and sustainable production of high-value natural products from plant cells. So yeah, I'm not going to read out these. So this is our sort of vision and our mission, and they're all around sort of people, planet, and progress. Our three kind of pillars that are reflected in our logo here. So plants are nature's premier chemists, able to synthesize a chemical cornucopia of structurally diverse natural molecules, which have been used in diverse industries, right? So, for example, you can have pharmaceuticals like Taxol, a blockbuster anti-cancer drug produced from Taxus baccata. You can have saffron, a cornerstone of the food industry, worth as much as its weight in gold, produced from Crocus sativus. And you've got things like morphine, for example, a key analgesic from Papyrus somniferum. And then in addition, you've probably got, 500,000–600,000 species of vascular plants alone. And only a few percentage of those have really been studied in any kind of detail to date. So there's a huge amount of chemical space still to be explored there. So lots of opportunity, I think. So yeah, these plant natural products are great, but then the problem often is accessing them. So getting access to these molecules can be highly problematic. So for example, they're very, very complex, structurally very complex. So trying to synthesize them is fiendishly difficult. And in fact, in the case of Taxol or paclitaxel, people have won the Nobel Prizes for actually synthesizing these things. So they're very complex. So trying to produce them with chemistry is often not a going concern. And even if it could be done, it's not really economically viable. So chemistry often is not possible. Of course, the obvious way is just from forest harvest. But again, these plants are not domesticated, so you can't grow them in agricultural settings. And the yield typically for some of these target natural products are very low. And the yields are not very robust as well, so they can vary dependent upon the season and the weather conditions over the year. So again, accessing from target plant species is problematic. And of course, if you're going into wild systems and collecting these plants, you're going to deplete these natural populations and upset the connected biosystems, which is obviously very important these days, as we're all aware of. So it's not a very sustainable way to access these molecules. And in addition to that, if you're harvesting from relatively exotic locales, your supply chain is long, so it's got a big carbon footprint. Your plants are going to be subject to disease, weather events, political instability, as we've seen has massively impacted supply chains over the last 18 months or so. So all these combine really to make accessing natural products from source plants often uneconomical, unsustainable, and also unreliable. So the Green Bioactive solution then is to use plant stem cell culture-based contained biomanufacturing at scale, uniquely enabling improved products for existing markets that already have committed budgets. So in this particular case, you can make accessing these molecules economical, sustainable, and also reliable. You can have a bioreactor in Dundee here to access these molecules. You don't have to access them from Colombian rainforest, and also world leading. So we've got proprietary, patent protected processes and products. Yeah, so I think increasingly the sort of biomanufacturing sustainability angle is becoming very important now for companies. So we've been able to definitely tap into that. So our processes have been shown by life cycle analysis in collaboration with University of Aberdeen to be very environmentally friendly. So I'm not going to go through all these, but for example, we can decrease the amount of land use and water use, sort of optimizing land for improved food affordability and also quality. So we set up Green Bioactives initially at the Roslin Innovation Centre in 2020, as I said, just in time for the pandemic. So there we've been using sort of early adopter funding to try and help scale our biomanufacturing platform and create and protect unique IP. We're kind of employing a business-to-business, product-specific co-development and out-licensing business model to grow the business, where Green Bioactives is retaining the rights outside of our customers' business-to-business needs. So, for example, the customer might be interested in agriculture space, but the molecule might have also applications in pharma, for example. So we can retain the rights in pharma, and our customers can then share the rights in agriculture. We're also discovering, making and selling our own natural products. So for example, hopefully by the end of the year, we'll have our first product on the market, which is actually a cognitive enhancer product from two natural products that function synergistically together to promote human and animal cognition. So first tested in animals and then clinically trialed and shown to increase human cognition by about 15 to 20% in clinical trials. So that will be coming out, at least in Europe, probably by the end of the year, and sold through European doctors initially in Germany and Poland, and then probably through into Spain and Portugal. So we'll also think we can grow different parts of the business. So obviously we have sort of a core technology that can potentially be used to grow the business in agriculture and food, for example, and also in healthcare. So we may be able to generate distinct businesses which may then have multiple exit opportunities. So we could sell Green Bioactives agriculture and food, and also Green Bioactives pharma, for example. So maybe that gives us multiple exit opportunities, which is good. So it's basically a platform technology, so we can go in, isolate these target cells. We normally go for the vascular stem cells, culture these up. We can then bioengineer them, so we've developed techniques to do that, and then scale up for biomanufacturing, and then provide our customers with the, either an extract or the purifying molecule to feed into their global supply chains. So yeah, we can potentially access all these different industrial sectors. So for example, things like ag and nutrition, sort of a $40 billion industry growing at 4.6%, or pharma, another 40 billion industry growing at 6.1%, and others. So it's a good core technology that enables us potentially to interact with numerous customers in different business sectors. Whoops. So as I said, we started up just in time for the pandemic, which wasn't good because we planned to go to trade shows and everything and get the message out there, which we couldn't do. But nevertheless, we were still able to get significant commercial traction. So we were able to interact with a number of companies, a number of multinational companies. Unfortunately, due to the agreements, I can't tell you the names, but anyway, these were in the US, in Europe, and also in Asia. So we were able to get significant commercial traction, which then helped us subsequently raise money, which is obviously an important thing to do. So this is just a sort of miniaturized example of how the technology can work. So in collaboration with an American agricultural company, global agricultural company, we have a project to produce a novel biopesticide for them, a sort of first in class from plant cell culture. So this is really being driven by the EU that want to ban 50% of all synthetic pesticides by 2030, although that's beginning to be kind of rowed back a little bit now, when the reality is beginning to sink in. But nevertheless, there's a big drive for benign, safe biopesticides that can be used. So, yeah, the American multinational came for us to use our technology to try and produce a molecule for them, which they identified from a Brazilian rainforest tree. So this tree only grows in the rainforest, obviously. You can't really go in and start chopping that down. So we were able to get access to the tree, and then we're able to isolate the vascular stem cells, culture those in media, get those growing in the lab. We're now beginning to bioengineer those cells. We've shown that they produce a target molecule. We now want to increase the yield of that molecule. So we're now beginning to bioengineer those cells. And then the plan, of course, would be then to scale those cells up and then feed into the global supply chains of this company. So they'll do the formulation and the sales and distribution, and we will just sell then the biopesticide. And this actually gives us 3 different value streams. So we'll be able to start generating revenue quite early because they'll need smaller quantities in order to do the formulation. So we can sell them that to enable them to get cracking on generating the formulation. We can also try and expand the use of this. So at the moment it's down as an insecticide, but it also has potential nematicidal activity. So we'd also like to explore that in more detail. Maybe we could do that with the JHI nematologist perhaps. And interestingly, this molecule is already designated safe food ingredient for the EU. So we can also potentially sell it outside of agriculture to a different customer, Nestle for example, to use as a food additive. So we have different ways to increase our revenue streams here. Yeah, so I guess the 3 USPs for the company are: 1, we can isolate and culture these vascular stem cells, which have superior properties, which I'll mention in a second. We also have various cell selection techniques that we can use to increase the yield of natural products without using gene editing or GMO transgene type approaches. And we also have good technology to be able to go in and identify the key sort of gene switches that control the production of these biomolecules. So most of these molecules are immune-related, controlled by immune-related pathways, so they're activated in response to perceived pathogen attack, and most of them are under transcriptional regulation. So identifying the key transcriptional regulators enables you to significantly increase the yield of these molecules by manipulating the activity of these key regulators. Obviously, if you can increase the yield, you can lower your production costs and then expand the market as well, and of course increase your profits. So we're very keen to do all 3 of those. So for example, in the case of paclitaxel from Taxus plicata, we estimate we can decrease the costs from about 20,000 a kilogram to 1,000 a kilogram using these technologies that we've employed. So yeah, some of the advantage of these vascular stem cells that we're able to isolate and culture. So these have specific biomarkers that we can use to identify them. yeah, interestingly, I guess because of their stem cell-like properties, they're actually actively dividing, and of course, the only plant cells that divide are found in meristems, all the other cells are just expanding. So these cells seem to be very well protected. So they seem to express high levels of plant defense responses and cellular protectant genes as well. So these cells are well protected. And I guess that's often true for meristems, which seem to be the only cells that can't be infected by virus infection, for example, presumably because they're very well protected. Yes, also they grow faster than the typical dedifferentiated cells that have been used typically in plant cell culture. And they seem to be more stress resistant. So I guess the figure on the right here just shows the classical dedifferentiated cells, or DDCs, growing in a 10-litre stirred tank bioreactor versus these vascular stem cells. So this is about 3,000 RPM, I think, with the stirred tank, and DDCs get stressed very quickly, whereas the VSCs are more stress resistant. Yeah, these cells also seem to grow either as singletons or as a clump of perhaps 2 or 3 cells. They don't form these typically very large clumps that you'll see with the classical dedifferentiated cells. So this enables them to have better access to nutrients, and they also seem to perform better as a consequence of that. And of course, then that gives you a homogeneous culture. Whereas with these dedifferentiated cells, you'll often generate a callus on a whole root or leaf, and it's a whole multigeneous cellular mix of different cell types. So that probably also adds to the less productive nature of these cells. So this is just showing their production of paclitaxel. So compared to these classical dedifferentiated cells from needle or embryo, these vascular stem cells produce significantly more of this natural product. They also seem to be much more responsive to elicitation. So often to elicit these pathways, as I say, they're immune related, you need to add in some kind of pathogen-associated molecular pattern, like chitosan, for example, or a yeast cell wall extract. Or in this case, we're using methyl jasmonate, which is a defense activator of secondary product pathways. So often these cells are much more responsive to the elicitor, and you get much better activation. And also, fortuitously, often the molecule is secreted into the medium more effectively with these cell types, which helps you with subsequent purification, because then often you can purify it from the media rather than having to break open the cells and purify it from there. So they do have significant advantages typically over your classical dedifferentiated plant cells. And but as I said, we've put some effort into developing technologies then to rapidly identify the key transcriptional regulators that can control these target pathways. So we can identify the key transcriptional regulators, and we're also looking at translational regulators as well now. So for example, we can add our elicitor, induce the expression of the pathway over time, do transcriptomics to get a whole expression profile, usually at times, short times after elicitation, and then look for transcription factors that are rapidly being activated to try and identify master regulators. So we'll get obviously a whole bunch of proteins, and then we can just use transient assays in our vascular stem cells of the relevant plant species to try and identify the transcription factors that are upregulating the promoters of the associated enzymes responsible for that pathway. So we can do that by constitutively overexpressing the target transcription factor with the target promoter hooked up to a luciferase reporter gene, plus a control luciferase reporter, vanilla luciferase under a constitutive promoter. So therefore we can test out rapidly candidate transcription factors to see which can drive the expression of target gene promoters. So this was just looking at the Tarsi promoter from paclitaxel below. And you can see that one transcription factor really strongly upregulates that particular gene. So doing that, and I can't give you all the transcription factors obviously because they're proprietary, but we can test out all our candidates for all the promoters in the pathway and then build up a network to see which transcription factors are required to upregulate or perhaps even downregulate the pathway. So in this particular case, there's a couple of transcription factors, B3, B4, that collectively are upregulating all steps in that pathway. So potentially those two transcription factors together could upregulate the whole pathway. And we can show, for example, that they bind to the target promoters. We can do cis element analysis to identify even where they're binding on the promoters using things like ChIP-seq, for example. So we can confirm promoter binding, and then ultimately, we can create transgenic cell lines or transiently express these transcription factors to show that they do indeed upregulate production of a target molecule. So this is just looking at paclitaxel production. So these two transcription factors, the wild-type copies, significantly increase paclitaxel production in combination. So having one of these transcription factors alone increases, but not by that much, but having them both together, you get a real sort of synergistic interaction and a significant increase in paclitaxel. If you make the mutants, then nothing happens if you overexpress the mutants. So, so it's good evidence that these two transcription factors in combination can increase your production of your target molecule. So we're able to do this with different pathways and hence increase the yields of our target molecules. So of course then we need to try and scale up our cells from lab scale to industrial scale. So we're now starting to do that. So we need the whole ecosystem of bioreactors to do this, going from our cryopreserved optimal cell line right through to 1,000 litre or ultimately 5,000 litre bioreactors. So we have plans in place to upscale. So we're currently around 50 litres, but we are looking to get up to 5,000 litre scale. And to do that, we're using real-time metabolomics guided by artificial intelligence. So we have collaborations with the University of Edinburgh where we can do real-time metabolomics within the bioreactor itself to follow key metabolites, measuring a few hundred different metabolites, and we can potentially measure thousands of metabolites offline. Then we can generate all the data, feed that into AI, and then use that to help guide the future scale-up as we go through these different iterations of bioreactors. So we're hoping that this process will expedite this process. So yeah, Edinburgh are the only guys in the world who can really do this, so we're kind of well placed to use this kind of technology to help drive our scale-up. So these are our scale-up plans. So we're kind of approaching stage 5 at the moment. Ultimately, we're going to get to stage 6 and 7, where we're going to need 1,000 litre, 5,000 litre bioreactors. So we'll need another round of investment for that. And perhaps these could be stationed. Well, they're probably not going to be stationed in Edinburgh. Probably off-site in Edinburgh, perhaps Scotland, but we've also been looking at sites in other countries. So I just got back from China last week, looking at investors and potentially making a production facility there. We're also thinking of perhaps India or the United States as well. So we have kind of an open mind on that at the moment as we move forward. But certainly we'll need a big production site somewhere because our customers are looking to us to produce molecules for them 2026, perhaps tons of these things. So we'll definitely need a big scale-up centre somewhere. So this is the team, myself, Yuan Li, R&D lead, who we've introduced already. So our new chairman, Thomas Nagy, he's a chemical engineer by training, which is great for our sort of scale-up work. So he was actually president for production at Novozymes, responsible for fermentation and scale-up. So that's a great fit for us. He was also then became president of Novozymes, and after leaving Novozymes, he was then the co-founder of the Danish Bioinnovation Centre and lead entrepreneur for that. So he's worked with stacks of startup companies, helping them get going, and has an extensive knowledge in the scale-up space, which is very useful for us. CEO is David McElroy, who has a PhD in plant molecular biology from Cornell. So a plant person, and worked for Monsanto and business development in DuPont, Pioneer, for example, and has co-founded a number of different companies in the US, and was also head of business development for Ingenza, sort of the UK's premier synthetic biology company for a while. And then on business development, we have Chris Meany, who worked for Cambridge Nutraceuticals for 20 years, going from its founding to maturation, and then he was head of business development for Primera Nutraceuticals as well. So Chris has got a lot of experience in this space. And then for operations, head of operations is Andy Hall Poncella. So he has a PhD in plant metabolic engineering from Oxford, and he was a co-founder of Biotangents, a spin-out from University of Edinburgh. Our CFO is John Craig, 20 years of corporate financial experience. And then on the production side for the scale-up, we have Bobby Bringy, who was actually co-founder and president of Phyton Catalytic. So they were the company that actually used cultured plant cells to produce paclitaxel. So he was the founder of that company, and I guess the only guy who's really scaled up plant cells to that kind of massive scale to date. So he's on our team, and also John Stevenson, who was responsible. So so both Bobby and John are chemical engineers by training. John was the person who helped Celtic Biorenewables scale up their bioreactor stationed at Grangemouth. And he also helped build Ireland's first whiskey distillery for 100 years. So he has a lot of experience in production and scale-up as well. And we also have a world-leading scientific advisory board to help us on our way. So I think it is actually a very strong team that we've been able to develop. So we're Green Bioactives. Our vision is to become the world's leading supplier of safe, natural and sustainably sourced plant-derived natural products, employing processes that help improve and protect our environment. So thanks for your attention, and I'd be happy to take any questions in the second part of the talk. Thank you. Yeah, go ahead.
So I have a couple of questions. On the technical questions initially, actually, okay. I've got quite a few questions.
Okay.
Right?
Okay, go ahead.
So the metabolomics for the scale-up screening.
Yes.
Right, so what are you trying to monitor there? Is it anoxia? Because I noticed you have lactate and succinate, right, as your two examples. Is that what you're looking to see?
I mean, we've got an open mind on that. So it's early days. We're just starting to do that. So so we just want to see what the chemical space is, as these cells grow in bioreactors. What are the key sort of metabolic inflection points that can tip them over the edge to make them unproductive, and then learn how we can avoid that, basically.
So is it a batch process then, or a continuous process?
It could be either. So principally a batch process, I think. Okay. So, I mean, potentially for some molecules it could be continuous.
And then the other technical question I had was on the sort of transcription factor screen.
Yes.
So you're identifying which transcription factors are regulating these pathways.
Yes.
Is that so you can then engineer the TFs, or so that or is that then to give you a hint as to what might elicit the induction?
No, looking to the transcription factors to engineer the pathways. Yeah.
So it will be engineered cells that you're using in the production. Yeah.
Yes. So could be gene edited, whether you say that's GMO or not. probably not, depending on what geographical location you're in. But then, yes, I mean, there could be transgenes also, yeah. So we could express those under conditional promoters, for example. Yeah.
I'll let someone else ask some questions, thanks.
Okay, yeah, Derek, go ahead.
A couple for you. What's the obvious ones? Why start the company? What prompted the journey?
Because, so just because companies were coming to me to do things in this kind of space, so I knew there was, you know, an opportunity to do something there. Yeah, so it's just an opportunity to do it. So I thought, well, yeah, I already know there's a lot of interest, so why not really just do it? Yeah. So that was a real driver for doing it.
Good. It sounded like there was possibly two streams to the business. So you talk about the company comes to you, I've got this molecule I want you to make for me.
Yes.
Is the other stream that you're creating your own initiative?
Yes, exactly. So yeah, I mean, ultimately we probably want to produce our own molecules, yeah. But in order to get going, you know, the companies coming towards us was very helpful, because then they gave us funding for co-development projects. So they give us a slug of money to help develop a process then to produce their target molecule. So they're quite happy for us to produce the molecule, you know, as long as it goes into their supply chains. So obviously they want to principally own the formulations and the subsequent applications, great. But we can own all the IP around you know, production, etc.
So I've got, like Rob, will finish maybe on a more technical one. It's all predicated on these VSC cells, the vascular cells.
Yes.
Presumably they're not as easy to culture from every species.
No, they're difficult, yes. Yeah, and it varies massively from species to species, yeah. Yes, so that's true. So we have a patent on that, obviously. Right. Yeah, so it depends on the species. So some species are relatively easy to do, some we haven't been able to do. so it's not a given that you'll be able to do that.
So there's one route, and this kind of goes back to old engineering.
But sorry, yeah.
But it's hard to culture from that species, but there's a sister species that might be easier, and then you just engineer that one up.
Yes, exactly. Yes, and I think as we generate more experience, so a key thing is like media composition, right? So I mean, once we understand more about that, so we're trying to generate a lot of information on media, and then we can perhaps use AI as well to help guide us to, you know, what media to use for a specific plant species. So that's kind of beginning to turn. Yeah, so I think as we generate more experience, we'll get better at those more recalcitrant species. But certainly, I wouldn't claim we could do every plant species. We can't.
In cell culturing in media was always termed the black art, really, isn't it?
Yes, exactly. It's a bit kind of voodoo. Yes, exactly. Yeah.
So do you see any toxicity actually ramping up these compounds? Like, are you seeing any evidence of toxicity from them? Yeah,
I mean, that can happen, yes, I would say. So so that's that in an idea. Well, I mean, most of them are either going to go to the vacuole or they'll be secreted, right? Ideally secreted, because then we can potentially, you know, take them off to remove any toxicity. So one of the things we're also interested in is looking at transport systems, for example, to help them get out of the cell more effectively to decrease the toxicity, or maybe you know, enzymes to get them more effectively into the vacuole. Yeah. So I think, yes, looking at those sort of detox systems will also be quite important for us going forward. Yeah.
So that, and I guess just go to one of my other questions, if you don't mind. So,
no.
So I mean, obviously, like, yeah. Well, I mean, I was wondering what your inputs. How are you dealing with your waste, and how are you dealing with your purification?
Yeah, that's
streams,
a good, yes, that's a very
particularly as you scale up.
Yes, that's a good question. I mean, at the moment, it's not a big deal, but once you get to 1,000 litres or 5,000 litres, yeah, that's going to be an issue. So, I mean, the plan would be to. So I think there, you know, a customer might want one molecule, but I suspect that, you know, those cells are going to produce a whole slew of different molecules. So we couldn't probably provide the customer with molecule A, but then we'll have A, B, C, D that could potentially feed into different markets. So that could be useful. As far as waste goes, we're kind of looking to break down those cells to generate sugars, which we can then, you know, grow our cells back on, so we can have some of a circular type of economy. Yeah. So we were looking to recycle or repurpose as much of that waste as we can.
And the purification, I mean, you know, these are still present in very small amounts, right, relative to the total bio.
Yeah, so downstream processing, you know, DSP could take some effort, but depends, you know, so it depends on the customer. So some customers, you know, they might be quite happy with just an extract that's enriched. Yes, other customers might want, especially farming, you know, 99% purity or something. So again, that'll be on a case-by-case basis. But yeah, so we're just hiring people now to do to start kicking off all the downstream processing. So we also need analytical chemists, downstream processing experts. Yeah. So we've recruited these kind of people now, starting imminently. So we're kind of going down that road. Yeah. So we're just so we're just hoping to sort of double our space soon, hopefully, to potentially have space to increase our production capacity. So we're kind of going down that route.
Good luck.
Thanks.
Any thoughts to do a Craig Venter approach and create a base vascular cell that you'll just engineer completely? I mean, is that an alternative?
Yeah, I mean, maybe that
A
might
platform that really works for you.
Yeah, I mean, maybe that might be possible. You know, so maybe we can have a cell that's really good at producing terpenoids, for example. Yeah. So that or another one for flavonoids. So that may well come, I think. I guess it's early days, yeah.
I think it's throwing all the data into the AI system, particularly the molecular data and metabolic flux and interaction data. That could guide you to something like that faster, I think.
Yes, so I mean, I guess mammalian cells, you think of Chinese hamster ovary cells, so they've had like 20, 25 years to really be fine-tuned and studied, and we understand a lot more about those now. And of course, they're being used as chassis to produce lots of therapeutic proteins like antibodies now. But, you know, plant cell culture, right in, you know, it's still virtually day one, yeah? So we know very, very little. So I think there's lots of things to learn, lots of IP to gather, you know, as we move forward in this. So as we kind of develop this as a chassis, you know. Yeah. So a lot of work to do, definitely.
So I was wondering if there was pathways that are sort of more tissue specific. So you were talking about saffron.
Yes.
You know, that you maybe couldn't get your vascular
Yes.
cells to make.
Good question. Yeah, absolutely. Yes, that could be true. Although, having said that, yeah, so often these secondary, well, maybe most of you know this already, but a lot of these natural product pathways can be in specific tissues. So saffron in stigmas, for example, yeah, or perhaps others in glandular trichomes, for example, yeah. So they can be quite specific. So, you know, whether our cells would then be able to produce those pathways is open to question. But interestingly, one of the customers, another multinational, British multinational, wanted us to produce a molecule that is typically secreted through glandular trichomes, but our cells produce it in reasonable amounts. So, yeah. So perhaps that may not always be the case, but certainly just because it's in a specific cell type doesn't mean our cells won't produce it. So we do have some evidence for that.
The other question I was going to ask you is whether your process does require light at any point, or is it all bioreactors?
It depends on the cells and what the process is, but generally we don't need light, yeah. Which makes things easier, yeah, and cheaper. Yes, exactly. But typically just sugars, yeah. But the sugar could vary. So
the question on Scaleup is really interesting, because the big problem we saw coming out lately was some of the guys I was mates with, Enough.
Yes, oh, they've done brilliant. Yeah, they've been very successful.
They did brilliant by going to the Netherlands.
Yes.
Scotland didn't have the Scaleup capability.
Right. Yeah. So they went adjacent to Cargill in the Netherlands. Yeah. So they can use all the kind of waste from Cargill to drive their process. Yeah. So yes, I mean that's something that, as I said, you know, maybe we have to go overseas also to kind of do this. So we're actively looking at that. But yeah, I think Enough were very successful to scale up their process. So Enough is a spin out from Stratified, yes.
Yeah, 3F Bios. Basically, it's quorum.
Yes,
And
exactly.
quorum's being finished. We just picked it up and tweaked it.
It's a different process to produce core, basically. Yeah, but they've been very successful in getting the funding to scale up and their production facilities. So they're finished now, I think. Next to Cardiff.
Is there any, are these processes can be used in cacti and fungi to look at psychedelics? And is there interest in?
Yeah, so our CBO is very keen on us doing something in CBD, you know. So yeah, I mean that is also produced through secretory trichomes, right? So whether our cells will be able to do that is open to question, but we'd be quite keen to look at that. And as you said, you've got a license, I guess.
Yeah.
Okay. So I don't know whether we could maybe collaborate on something.
Chat for this.
Yeah. So we'd definitely be interested in seeing whether we could get something going there, because there's a big demand there. Yeah. And I guess it's probably a lot easier for us to just grow the cells than it would be to grow the plants, which are, require all kinds of security and issues. But if we can just get the active molecule from culture
you
plant
get
cells,
closer to a more pharma-friendly version, because pharma is still worried about cannabis because of the multiple different compounds.
yes, quite, yeah.
If you're producing one or 99% enriched one, they might be much happier.
Yes. And I guess as we know more, then there'll be other compounds that will be discovered that also need to be produced. So that's a growing market and something that we should consider. So yeah, we'd be quite keen to try and do something in that area. So yeah, maybe get some collaboration going.
I think you'd mentioned actually fungi. Have you tried fungi cells in your systems?
We haven't. No, we haven't. So I think we've got.
You've got enough.
I think we've bitten off far more than we can chew with just plant cells, let alone getting into another kingdom. Yeah. So I think we'll probably concentrate on the plants. Yeah.
It's for another company.
Yeah, absolutely. Yeah, that's probably for another company. Yeah. So the plant cells is more than enough. Yeah, yeah. Especially with different products, you know. So often it's like Phyton, they had one product. That was it. Concentrated on that and got it out. And for the last 25 years, that's all they've done. Yeah? So we're trying to do a bit more than that.
And as we discussed earlier on, I mean, the obvious exit strategy is if you get the platform that works in several compounds, someone just buys the company.
Yes, exactly. Yeah, yeah. So probably one of the big multinationals that we're working with. Once we get used to working with them, become an integrated part of their supply chain, then it would make sense for them to just buy us. But as I said, maybe if we can develop different areas, they can just buy Green Bioactives agriculture, and we can sell Green Bioactives pharma to VSK or something. So we may be able to have multiple exit strategies, which would be good. Double our money, triple our
You can
money.
do the next stop from the Bahamas then.
Well, yeah, maybe we can often buy a small island in the Caribbean. I don't know. We'll see. I'm not counting my chickens, considering that most companies will fail. So yeah, but we'll see. We'll try our best. Yeah, that's all we can do. Yeah. So we try. Yeah, but I think we do have a really good team. Yeah, so we're making headway. So far, so good. Yeah.
Okay, well, thanks for that, Gary.
Thank you. Yeah,
Appreciate it in
thanks.
a normal manner.
Thanks, Gary.